So now we've just finished checking our PCR product and our agarose gel, and it looks like they're good. So the next step is going to be restricting both of our PCR fragment and our plasmid. In this case, the plasmid we're using is PQE31. So the enzymes that we're going to need for our restriction digest actually have to be stored at minus 20 degrees Celsius in our freezer. Those are maintained in the lab next door. So I'm going to walk back and show you where we keep those, so you'll be able to go and get them yourself when you're ready to do your restriction digest. We're now in our preparation lab, and in this freezer, right up top here, there is a blue and white ice box. So on a little bit, it's got cloning listed on it, and all the various frozen elements, enzymes and buffers and things that you'll need for cloning are located in this box. So to perform our restriction digest, we need a couple of elements. Obviously we need the DNA we're going to digest. So for one of my reactions, this is going to be my PCR sample. And then for my other reaction, it's going to be the PQE31 plasmid. And that's located in my ice box. So you can see that's labeled right there. Now this has to thaw, and so we are going to set this out in our rack so it will thaw for a little bit. There's also going to be our buffer that we need. It's buffer number 4 from NAB. There's several different buffers that work with different restriction enzymes, so we're using NAB 4. And we are going to place that in our rack to thaw as well. Our two enzymes, KPN1 and Hindi3, are enzymes. They're stored in glycerol and they don't need to thaw, and we're going to leave them in the rack to keep them cold. So we're going to work with our PCR fragment first and do that digest because it's already cooled off. So I need 35 microliters. And I need to pick up, my digest is going to be in a 1.5 milliliter Eppendorf centrifuge tube, or an EPI for short. And I'm going to pull off my sample. And let's see. Do this one. I'm going to pull off 35 microliters. Pipette that into a centrifuge tube. Next thing I'm going to need is my buffer. I need 4 microliters of buffer. And now I need to add my two enzymes themselves. So I need just about half a microliter of each. I'll tune this to half a microliter. Now I'll do first the KPN enzyme according to my recipe. And so the KPN enzyme just takes a little bit of the pipette tip. That's half a microliter in my pipette tip. And on this one, I need to make sure that my pipette tip goes all the way into the solution so that I make sure I am delivering all of my KPN enzyme in there. With small volumes, it has to go inside the solution. Now do my Hindi3. Make sure I have half a microliter in there. And then again, I'm going to place it all the way into my reaction. Make sure it's all in there. Now I'm going to take up my orange pipetter again, and I'm going to mix my sample real well. If you don't mix, the KPN and Hindi3 enzymes are stored in glycerol, and they'll actually go right to the bottom of your sample.
When you're like applying for graduate school, like they fly you out there and they'll
tap it and So that's one of our reactions. And before I go any further, I want to make sure that I label this reaction. And so for this one, I'm at station number 1. So I'm going to put, I'm section 2, station 1 on here. And this is my PCR digest. I can't fit digest all the way, so I'm just going to put DIG, or dig. That's done. So now I'm going to label this one as section 2-1. And this one is PQE31 digest. And now that these are both prepared, I'm going to grab both reactions and I'm going to put them in the 37-degree incubator for the reaction to take place. So from my station, I'm just walking around to the back of the classroom. And this is our 37-degree incubator. Just open them up. There should be a rack in here. I'm just going to pull this rack out, put my 2 samples in, slide it back in. Now if there's not a rack in here, then they can be found over on the left side of the classroom. And I will leave these in the incubator for at least 4 hours. I can leave them in for longer, I can leave them in as long as 16 hours, but sometimes you'll get more degradation. So probably the ideal time is between 4 and 8 hours for the restriction digest. Now while our restriction digests are incubating, since this is a community lab, I want to make sure I come back and I need to take my reagents and replace them. So I'm taking my buffer and I'm going to put them back in my ice box. I'm going to take my PQE31 vector and put it back in the ice box. And I'm going to take the rest of the PCR samples now and put them back in the ice box so that they'll be ready for the next team that comes in.